So evolution, molecular evolution, and that's where the concept of gene shuffling and direct evolution comes into picture. We can design the molecules in such a way that they are able to function in a way which nature has not thought of. Then microbial interaction. This is one of the aspects, as I mentioned in the morning, that microbiologists work on microbiology, microbes. Botanists work on plants, but they're interactive because in nature they do not live in isolation. So even microbe interaction has not been paid much attention. Forget about microbes, animals, microbes, plants. So interactive association is very important aspect, uh, and and that's what is being paid attention to these days. And then next, uh, focusing on extreme environments. It may be salinity, it may be temperature, it may be combination of more than extreme conditions. There are many microbes which are able to live under combination of different extreme conditions. So in that way, they are evolutionarily relaxed because they are able to live under the conditions which are not normal. And because of that ability, they are very important. They are in focus in search for microbial or any form of life on different planets. So that is very important from that point of view as well. Now the further debate is between individual organisms, metagenomics, and micro. For a long time, we have been focusing on individual microorganisms. You isolate bacteria, you study them in the lab, but the way they will behave in lab will be entirely different than the way they, or pharmaceutically, they may be important. They have been used for producing antibiotics, of course, very useful, but what they behave, what they can do in nature, very limited knowledge we have. So conventionally, we have been isolating microorganisms. We study them, cultivation, their morphological features, their cell morphology, and also certain behavior. For example, whether they are able to grow in organic solvent or they are unable to grow. A similar kind of unusual conditions, whether they are able to live or not. So these are some of the things we have been doing for long. Then what are other criteria of diversifying or judging the diversity of microorganisms? For example, cell chemistry. Cell chemistry is one of the criteria. For example, cell membrane, cell wall, their components, for example, amino acids, sugars. We have seen over the last 20 years that if you analyze these molecules in cell wall, it can be a good criteria to judge the diversity. We can convert into clusters, and these clusters can be very useful in. So not only molecular aspects, but even non-molecular aspects can be useful, even in case of microorganisms. Then molecular aspects. Molecular aspects, for example, 16F ribosomal based analysis are simply using different primers. Using different primers, we can generate. There's some examples that I'd like to show here. This is the microorganisms isolated from two different saline habitats. You can see using four different primer sets, we can generate different profiling. So using number of primers. So on the basis of the amplification pattern, or whether the amplification is taking place or not, on that basis we can diversify microorganisms. This we have seen in case of actinomyces. Actinomyces are difficult to grow, of course, but even in case of actinomyces, we have seen that this parameter can be very useful. And based on those parameters, phylogenetic status can be established. And the second panel actually describes non-genetical aspects. For example, biochemical parameters, sugar utilization, morphological characteristics. Then those parameters which you can convert into binary functions, binary matrix, presence or absence, plus or minus. So based on that matrix, we can generate kilograms, and based on that, we can generate. And sometimes these kilograms may relate to kilograms or may not relate. So This means that even non-genetical parameters are very important in case of microbes as well. Of course, animals and plants, these parameters are very important. But even in case of microorganisms, where morphological variations are very limited, but these parameters can be converted into a matrix, and that matrix can be used. This is just importance of non-genetical parameters. There are certain basic features of these microorganisms from saline habitats. Some of the features, for example, they produce array of enzymes, hydrolytic enzymes, protease, amylase, and so on. And these enzymes, these organisms can grow under variability of the pH, salt, temperature, as well as their enzymes are they're capable to act under different conditions of pH, salinity. Even though the organism is not thermophilic in nature, this is very important aspect. Even though the organism is not thermophilic in nature, but their enzymes are thermophilic. So the enzymes are able to function at alkaline pH, they are able to function at high temperature, they are able to function at high salt concentration. So many of organisms, they have this capability. And they can be expressed, and the effect of salt can be, so the enzymes forms halophilic can be expressed in E. coli, and many such examples we have. This is one important aspect, that the enzymatic parameter, what is shown here, this effect of temperature on the profiling, that in the presence of salt, the enzymes, this is a particular enzyme protease, the temperature profile, there is a shift of 30 degree. So 30 degree shift from you can see from 45 to 80 degree. So that is salt dependent, because if you remove the salt, they are unable to. Now this is another example, the same thing. Then another parameter, most of the enzymes, as you are aware, as students in biochemistry, you know that if you treat them chemical denaturants, for example, urea, 6 molar urea or 8 molar urea, okay, so what will happen? Most of the enzymes or proteins will denature, okay, they will lose their activity. But many of these enzymes from halophilic organisms, they are highly resistant to chemical denaturation, and this is a general phenomenon that we have found in many cases. Later on, we found that this property is not independent. This property is dependent on salt, because if you remove the salt, they become very sensitive. They will denature as usual. So this is another property that we notice. Now, kinetically also, this is just to demonstrate how kinetically the enzymes can behave in the presence of salt. Normally, if you increase the temperature, most of the enzymes, they will lose their functionality. But that process can be arrested by addition of salt. That has been shown kinetically as well. Catalytic efficiency can be retained in the presence of salt. Some other properties, of course, they are there, but I just noticed, highlighted some of the key. One of the other extremity is the organic solvents. Many of these organisms, so as I mentioned, that these organisms, they have multiple ability to function under a number of extremities. So one of the extremity is solvent, and one of the extremity, their ability to grow in chemically defined medium. And the synthesis of enzyme is governed by carbon-nitrogen ratio. And this is one example to demonstrate that they can grow in the presence of amino acid. Amino acid can be used as a carbon and nitrogen source. So without supplementation of any organic component, they can grow very well and produce enzymes. Then cloning and expression. Many of these enzymes. So our purpose was to demonstrate whether we can clone and produce E. coli, which is a mesophilic bacteria, Number of genes we have cloned from halophilic bacteria as well as actinomyces, and we have expressed in E. coli, and the expression level and effect of salt, and whether the enzyme retains functionality. These are the aspects that we discussed, and we have uh, published in many papers. Now, another thing that these habitats, they have very novel organisms from diversity point of view. So far, I have been discussing certain key features of properties reflected by organisms of halophilic nature from saline habitats, different kinds of saline. Saline habitat doesn't mean that Gujarat is longest coast, 1600-kilometer long coast, and longest coast in India. But that is, see what is not only habitat. Salt pan activities are there, and many and saline soil is there, desert is there. So everywhere there is salinity or combination of salinity. So there are very novel organisms which we have found. Some we have published because we have been able to establish their nobility. We have proposed the species, but there are many which we are still in the process to establish their nobility in terms of taxonomic proposition. Then now I will come on interaction, the plant microbe interaction. Plant microbe interactions, the plant growth promoting microorganisms. They help plants in different ways. Some of the ways are direct ways, for example, nitrogen fixation, transport, other things, whereas some are indirect, for example, cytoplasm. So these are things that you study in your syllabus as well. Now, stress. How these microbes or interaction of the microbes can be useful to the plants in combating those stressful conditions, for example, salinity, as well as pathogenicity, for example, fungal growth. So these microorganisms, they are very much capable to help plants to fight against pathogenic conditions. Okay. There are many strategies. Of course, there are many strategies at biochemical level, at physiological level, at genetical level, which are used by different plants in association with bacteria. Okay, and they help plants in fighting against fungal disease, fighting against stress tolerance, or both. Halophytes, number of halophytes we have isolated from saline habitats, and we have isolated the microbes from their root, Rhizospheric, and we have studied. Of course, we have studied in lab with the isolated and then properties of those organisms. And this is just a reflection that how important this aspect is. Salinity tolerance or salinity is a big problem, and this problem is very detrimental in terms of productivity of the plants. For example, this is what is shown here. The salinity, a significant portion of the cultivable and irrigated land is affected by salinity. And even though there are certain chemical remedies, but these have very limited use, and usually they have adverse impact on the soil health. So then comes this strategy of biological system, for example, microorganisms helping plants to combat these problems. Again, there are two measures in this interaction. We can study microorganisms, we can isolate and study them in isolation, or we can study the entire microbiome using metagenomics aspects. Now, focusing on some aspects of cultivable rhizospheric bacteria. That rhizospheric bacteria, many people have worked on rhizospheric bacteria. They have isolated, they have studied their properties, and they have tried to relate to their significance in plant health. And they have isolated. They have seen how does it affect the seed germination, how they affect the different agronomic properties, root length, and so on. Now, so just there in the literature you can find these are some of the examples of literature where people have studied different groups of the organisms, have been studied, and different properties. In the third one, different properties which are related to PGPR properties, and the number of properties, for example, their ability to produce enzymes, their ability to enhance or activate the hormone production, Number of organisms isolated from these halophytes as four halophytes from coastal Gujarat, and you can see the number of microorganisms in terms of their number as well as in terms of their diversity. It varies among these, although they are from similar habitat, but there is a variation in terms of their number, in terms of their properties. This is also there in terms of the properties. The four groups of microorganisms isolated from four different halophytes, four different plants. You can see that profiling which is different. For example, in this case, ammonia production. Similarly, many other properties, for example, this is a phosphate solubilization. So any property you take, you can see the variation and diversity of the microorganisms from the plants from a given habitat. Enzymatic, the same thing is the enzymatic profiling. Now, the production of enzyme is under salinity because we are talking about salinity. So whether these microbes from halophytes, they are able to produce enzymes at salinity. So they are able to produce at salinity. We have seen up to 200 millimolar, and beyond that, there is a detrimental effect. Antifungal activities. Antifungal activities, because that relates to pathogenic status, whether the microorganisms which are part of Hydrospheric, they can help plants to combat this problem. So a number of organisms have been studied, pathogens have been studied in case of their pathogenic ability, and how these microorganisms, bacteria, can help to reduce this, to inhibit those pathogens. Very effective cases, and we'll see. These are some examples again from literature. I would skip this here. Now, this is just an example to demonstrate that at different salinity, the impact of these microorganisms to fight against fungal pathogenic status. These microorganisms very effectively help plants to reduce the fungal pathogen. They can inhibit the fungi causing disease in plants, and so that is at different salinity conditions. Fusarium, and before that it was Aspergillus, one fungal pathogen, Fusarium, Alternaria. These are different 4 or 5 fungal pathogens. Similarly, as I mentioned in the beginning, certain agronomical properties also. These bacteria, they are very helpful in assisting the plants in terms of the root length, and seed germination at different salinity. That is important because the idea is that whether these plants can grow well and can fight against pathogens, as well as their productivity cannot be reduced. So this is what this is parameter, economical parameter, seed germination, and this is just to demonstrate that number of organisms they have certain common properties in all. For example, this Venn diagram actually shows that at least some common properties shared by all bacteria isolated from different plants. Now, proteomics and perspectives of PGPR, mostly based on literature, gene expression affecting different aspects of metabolism has been studied. Then small RNA, how the small RNA can be helpful in combating the salinity as well as fighting against pathogens. Now coming to metagenomics and hydrospheric microbiome. Now, if you talk of metagenomics, the first thing is to extract the DNA. The first thing is to extract the DNA from the environment, because when you talk about metagenomics, you are taking the entire genome of that environment, and that entire genome is represented by majority of the microorganisms which we are not able to grow. Okay, so that's why. So extraction of the genomic DNA from environment is very important. That is the first stage, the quality and quantity of the genomic DNA, there is no set method actually for every habitat. We have to develop methods, and number of methods we have developed, and we have modified so that we can get good quality of DNA in good quantity. This is just one example of saline habitat. You can see that these saline habitats, two different saline habitats. One is Alan, which is a anthropogenically more disturbed area. One is pristine. So you can see the composition. You can see the microbial composition, how distinctly it varies at phylum level. Similarly, at genus level also there is a variability which reflects. Of course, majority of the microorganisms they are reflected as unclassified, but still the whole profile actually tells you about the diversity in these two different habitats of saline nature. Now, rhizosphere microbiome, how important it is in the plant ecosystem. As far as I mentioned at the beginning, people have been working on rhizospheric microorganisms for long, but in most cases, they have isolated, studied in the lab, and maybe application, but entire community has not been paid attention to, which is being paid now. The entire microbiome, rhizospheric. Of course, there are many other habitats. For example, gut microorganisms, gut metagenomics. So we will not talk about gut, but our particular emphasis is on this rhizospheric microbiome. So limited information is available. We have limited knowledge about the microbiome of this, and this is microbiome under stress. What is important is that if you grow the plants without any stress, either in nature or in controlled conditions, so the microbiome, the composition of microbiome is very different. So it means that under stressful conditions, the composition and dynamics of the microbial population changes rapidly. And that is the important aspect, that under which conditions, because these microorganisms, they are beneficial microorganisms. So in the condition of any stress, there is a recruitment of beneficial microorganisms, and these microorganisms, they help plant to counter these adversities. Now, there is a concept of core microbiome and minimal microbiome. Core, because for a function, there is a minimum number of microorganisms which are required. Means if they want to help plants, if they want to help plants in stress salinity, if they want to help plants in fungal pathogenicity, so there should be a minimum microbiome. Minimum means not only in terms of the taxonomic diversity, but in terms of their functionality. Okay, so how much minimum function is required? Because what is the reason? What is the concept? That if there is a redundancy in the function, for example, there are 20 microorganisms which have the same property, so that would not be desirable. So the functional redundancy should be reduced. So our focus should be on functional diversity rather than taxonomic diversity. If the same range of functions can be performed by 10 microorganisms, why should we go for 100 microorganisms? So the focus is to reduce the taxonomic diversity, or number of different types of microorganisms which are taxonomically different. Our purpose is to have functional diversity, or the minimum number of functions produced are reflected by minimum number of microorganisms. Because if there is a redundancy, then that will go antagonistic. Because the large number of microorganisms, larger the number of microorganisms, larger the chances of antagonism. And our purpose is to reduce that redundancy, and we want a minimum number of functions scattered by minimum number of microorganisms. That is the concept of core microbiome and minimal microorganisms. That is the area where people are focusing. That and that can be used artificially also under in vitro conditions or under controlled conditions. Like gut, there are many concepts of therapy where there is a gut microflora. It can be changed, it can be inoculated, it can be manipulated or maneuvered. Similarly, in this case. So the concept of minimal microbiome and concept of this concept of minimal, so that the functional redundancy can be reduced. So our focus is not that how many different diverse taxonomic types of bacteria are there, whether minimum number of bacteria are able to function certain number of properties or provide certain number of support. This is actually just academically, I just came to this recent paper, and this is what they had reflected, that as soon as there is a pathogen comes, then beneficial microorganisms, they make a shield actually. There are certain signals. We'll talk about those signals. There are certain signals are sent, and that those signals actually initiate that these beneficial microorganisms, they should come now in action, so that these pathogenic bacteria, pathogenic fungi, they can be pushed out. This is a very interesting paper, which is just published this month only. And arid, this is one particular type of arid desert, and arid desert where microbes actually in stones are they go inside the stone as a strategy to protect themselves against different kinds of adversities. Okay, so what they actually found, what is important, that because of fluctuation, because of heavy rain, the whole environment changed. The environment changed means their salt or osmotic conditions changed. Because of heavy rain, osmotic conditions changed, and that change actually led to significant change in the microbiome. But in due course of time, there was restoration. Few months, maybe 1 year, I'm not sure. There was restoration of the microbiome. But what they found, or what they concluded, the restoration of microbial forms was not exactly the same, or was not near to the same as it was earlier. Whereas the function was almost similar. The microbial force or microbiome changed, but function did not change significantly after the restoration of that particular condition. That is very interesting, because we have been taught, conventional wisdom tells that the transcriptional landscape or transcriptional box that we have in mind, that is generated by your genome. So we are saying that microbes have changed, the genome has changed, but transcriptional box did not change, and that's why the function was restored. A very interesting concept, that without changing the transcriptional box, even though the microorganism change, consortium change, our population dynamics change, but the function was restored. Okay, so that was just one example, interesting example in this theme. Now, functional amplicon. This is just a technical, that the functional amplicon, because for every conditions there are certain conserved domains. For example, if any pathogen comes, any salinity stress or any other type of stress comes, then there is a particular domain, particular metabolism or particular conservatism in bacteria, which comes into the picture, and it helps the plant. So identifying those domains, identifying the conserved domains and specific metabolic pathways which are required to defend the plants under these conditions. Now, these are some patterns that we applied and is being considered on the bacteria which have these PGPR traits, and we consider that they're very useful. We have demonstrated, and it's being considered by the agency. Then, to conclude, PGPR Microbial is very important for long. People have been working. That uh, but the entire community has not been focused in a larger context. So that is the focus, and with the help of metabolite identification, and with the help of bioinformatic tools, docking, with the help of docking, we can establish that these metabolites actually they are important to fight against plant pathogens, against salinity, and then we can identify these markers and we can proceed ahead. So there is a need to focus and intensify the research on microbiome, rhizosphere microbiome. Few interesting things I would like to share with you recently. One of the Nobel laureates, Professor Sidney Brenner, and who actually worked on C. elegans, and what he observed that in today's time, while we are generating huge data are being generated, but the purpose of science is not to generate data, to generate ideas, to generate thoughts, to generate hypotheses, but that is not happening. So that should happen. The ideas should be generated, thoughts should be generated. And that's observation by one of the Nobel laureates. Another observation is an article from D. Subramanian, who is a scientist, very celebrated scientist in the University of Science. And the language of the literature, scientific literature, is getting more and more complex, earlier, even people from not from scientific domain, they can understand the language. With time, the language is getting complex, so that people outside, even people within the scientific domain, sometimes it's difficult to understand. I'd like to acknowledge my team, and this is a research team over the last 2 decades. Many of them they are now faculty members, and they work in our department. They work as a women scientist, as a DSTM scientist, and now they are different places. And plus PhD students, I would like to acknowledge their contribution, because whatever we are able to achieve, that is because of the young team, and without their help, their spontaneous involvement. Because if it is done by force, then that is different thing. But spontaneously, if they are able to involve, that is very important. So our PhD students, about 25 students, which have contributed to different areas of our research. Financial assistance from different agencies, BDT, UGC, BST, Ministry of Earth Sciences, and GSBTM is a Gujarat government's own BST actually. And our own university, Saurashtra University, it helps us in different ways, including financial assistance for projects. Our research collaborations, active functional collaborations with Professor S.K. Khalid from IIT Delhi, with Professor Sanjay Kapoor. We had a joint multi-institutional project from BDT, and we worked together for 5 years. Still, we are continuing that from Delhi University South Campus. Then my other collaborator is Professor Yusuf from National Food Research Institute. Now he's at Delhi University, where I worked for 2.5 years as ASPS fellow. Griffith University, Brisbane, Jawaharlal Nehru Technical University in Hyderabad, and Central University in Hyderabad. These are our functional collaborators, not just our friends, but functional collaborators. We have research projects together. We have research projects together. Some publications, just I'll skip these things. Our research team, as students come and go, they spend sometimes 4 or 5 years. The dynamics of students change. So this is just to demonstrate some of the dynamics of students. The upper we have 4 PhD students and 4 MSc students currently working. under the project sponsored by UGC in this year of faculty. This is the slide that I always share. Particularly for our young friends. Mostly people do not ask questions, or they do not express their opinion. If the questions are not asked, if doubts are not raised, no new knowledge will be generated. And this is a thought given by a physicist, a Nobel laureate physicist, Feynman, the theoretical physicist, and I fully agree with that. More and more questions from younger. If you do not ask questions, if you do not raise doubts on certain things, new knowledge will never generate. Okay, so that's why please be involved, ask questions, even at the displeasure of your teachers. I always say that कभी-कभी अगर वो थोड़ा annoyed हो जाते हैं, तब भी take the chance and risk, but you must get involved. And these days, last 2 years, we are online education. It seems that we are talking to screens. So please get involved, ask questions, and not only questions. You can suggest few things, you can feel certain things, and share that. So this is related to the rate of extension. That is, of course, this is more related to this is one of the aspects that we discussed in the morning. The rate of extension, which is increasing very. This is just one example. I came across as a just a reader that since 1900, the rate of extension of plants, almost three species of the plants have plants getting extinct, and that rate is actually 500 times greater than normally should be. So that is the point of concern. That is just a glimpse of greater run of Kutch. I think some of you might have visited, and that this is how it looks like in dry season, in summer. I thank you all for your attention. I wish that you have some questions, and sir, I hope that I've been able to complete in time, and I hope that some questions. I'd be happy if you can share your opinion. You can ask something. Asking question doesn't mean that I know everything. I may not be able to answer your questions. Then we have another expert from plants, and they will answer if it is related to plants, or taxonomy, or physiology, or ecology. But it would be nice to hear from you.
Inquiries from anyone? Then press discussion.
As I mentioned in the beginning, most of us, we have worked in isolation. In isolation is, for example, if you are working with bacteria, you are only working with bacteria. But that is only limited story. The full story is the interaction. So unless we focus on interactions, then we would not be getting full view of their role in ecosystem, and how can we use them for maneuvering that, not manipulating, but I would say that maneuvering the ecosystem, so that without disturbing ecosystem, we can restore, we can use certain biological potential for our benefits. Oh, yes.
So you have done a lot of work on this population innovation. Just I want to understand that how they variations in
our rate of mutation in microbial system, I think, is greater than smaller the genome. Perhaps like the viruses we are talking about, the variation is much extent of variation is bacteria much larger, but the rate of variation actually is affected by internal as well as external factors. For example, the salinity or any other type of are their own. The certain genomes are more plastic, are more they are more able to change themselves. So there are many factors, genetic factors, biochemical factors, as well as environmental factors, which decide. But in general, the smaller organisms larger the chance of getting them changed. जी हाँ in case of bacteria they yeah there is variation, sir, because many properties that we mentioned, that even taking example of saline habitats. Saline habitats, taxonomically also they are very variable, phylogenetically very variable, means their genomic status is variable, even they are from the same habitat. So certainly the same habitat, even same gender, their genome structure can be variable. And that is also reflected in their biochemistry, their molecular structure, and their other properties. So certainly, within the same habitat, there is a variability in terms of their ability to get manipulated. I think. Yeah. This is simply query from my side. I just यहाँ पर आ जाइए नहीं वो ऑनलाइन जो है ऑनलाइन इट्स गोइंग ओके ओके thank you very much for your nice talk and informative talk. Being a microbiologist, I have also worked on some metagenomics. Since I have worked on the extremophiles and mostly the halophilic salinity. What is your opinion about the metagenomes of a particular habitat, where we get the bacteria, fungi, xenomyces together? But in the total genome, it will have the genome of bacteria, fungi, xenomyces. Only the functional aspect. Functional aspect. But for taxonomic also we have very specific primers. So based on that primers, we can distinguish. For example, in a given habitat, if you want to, like what we showed, we just focus on. Hello. It will amplify only from bacteria. Yeah.
Sequences. I'm talking about.
So functional metagenomic, of course, then you have very specific. But even in taxonomic distinctions, up to certain extent, you can distinguish. Using specific examples where we have a conserved sequence of bacteria, xenomyces, and fungi. Fungi, we have not worked on fungi. We work only on bacteria and xenomyces. So among the bacteria, of course, because in prokaryotes we have used 16S, but 18S we have not. But that can be conserved. Convergence can be possible. We have plenty of data. But that is very interesting in the sense that over the period resolution which has taken place, many of the genes have been acquired from external sources, horizontal, rather than just what. So during the millions of years, and those parts they become part of that particular organism. Like in E. coli, significant portion of E. coli is derived from other sources. Similarly, I think it will be true for other microorganisms. Thank you, sir. Thank you. Thank you very much. But I would like to students.
reproducibility of the genes from some other as we been to
Viruses, hallucinate.
they also definitely have some genes from
Because microbial system, particularly bacteria, uh, because like there are three modes of genetic exchange: conjugation, transformation, and transduction. So one of the method is through viruses. See, so virus certainly plays important role in this evolutionary process. And that's mostly interest specific. Yeah, yeah. Otherwise, it will be degraded. If it is not complementary to the, it will be degraded. So unless it is integrated into the chromosome, it will not bring the variation. Thank you all. Thank you all once again. All the best to you. Best of luck.
Thank you, Professor S.P. from Saurashtra University, for a wonderful and very illuminating talk. You have summarized very, very nicely your experience of your life working on microbes, especially strategies to encounter salinity stress. And that's a very, very important area of research for a large piece of land of our country where we have salinity stress, especially in western reaches of the country. This your research has certainly guided and guided to resolve some of the problems. You have also counted a lot of milestones which have occurred during last 60, 70 years in these areas of research. And also, one of the highlights was that you have two, especially younger generations, told that what should be the future course of research, in which direction the research is moving, and especially the metagenomics and microbiome, the phylogenetic and phylograms, and then this concept of minimum microbiome, the threshold level which is required to be diverse and functional to control the various mechanisms and the productivity issues has to be addressed. I would say here the younger people like research scholars, especially UG and PG students, I think you should be a little bit more interactive and be determined that you ask some questions. Don't worry about what people will tell about your questions, because most of the path-breaking researches have come out of asking foolish questions. Okay, so be proud of asking something which nobody would really expect. Okay, so I would like to expect from you some more questions. Thank you very much, Professor Singh, once again. And we move on to the session with the next. So please call on the next speaker in the line.
ये बार बार डिस्कनेक्ट हो रहा है उसके प्रोसेस रिजोल्यूशन का प्रॉब्लम जा रहा है सर एक सेकंड
Hello, hello. इको हो रही है? We will begin with paper presentation. Each speaker will be given 5 minutes plus 2 minutes additional. The first presentation, ethnomedicinal uses of 6 wild cucurbits commonly occurring in lower Assam region of Northeast India. Anand Bakshi, are you present?
Yes, I am present.
Hello.
Hello.
You're not
Am I audible?
Hello.
Hello. Am I audible?
Yes, you are audible. Please, can you put on your camera and start sharing your presentation?
Okay.
Hello.
Yes, I'm sharing my screen first.
Hello.
Hello. Is it visible?
Hello. You're not audible. Can you speak a little louder?
Okay, okay. So now am I audible?
Yes.
Okay. I'll give you a start. Please wait for a minute. So first presentation, number 3, ethnomedicinal uses. Yes, sir. He has started. Yes, sir. Anand Bakshi. Yes, he is online. Yes, you can start.
Okay. So just clarify if my slide is visible now. Okay. So a very good morning, respected members of the panel and delegates present in the symposium organized by Guru Ghasidas Vishwavidyalaya, Chhattisgarh. I am Anand Bakshi, a research scholar in the Department of Botany, Northeastern Hill University, working under the supervision of Professor Uma Shankar sir. So today I am going to discuss about the ethnomedicinal uses of 6 wild cucurbits commonly occurring in Lower Assam region of Northeast India. So, introduction. As you all know, ethnobotany is the study of relationships between plants and people, and today's presentation is based on the ethnomedicinal plants. So, ethnomedicinal plants form the basis for many pharmacological research. So, the study and documentation of ethnomedicinal plants is of very much importance. A number of therapeutic drugs have been developed and marketed on the basis of ethnomedicinal leads. Besides, wild edible plants contribute greatly to the nutritional well-being of many rural communities And agrobiodiversity also includes wild relatives of cultivated plants, and they are useful in crop improvement. Therefore, documentation of traditional knowledge and conservation of wild germplasm is of great importance. Coming to the family Cucurbitaceae, it is a family of tendril-bearing climbers, mostly herbaceous annual vines or perennial lianas. They are predominantly distributed in the tropical and subtropical regions of the world, and the northeastern region of India is very rich in cucurbit diversity with more than 73 taxa. The family Cucurbitaceae is well known for its commercially important crop species. However, high nutritive and therapeutic properties of many members make them excellent source of herbal medicine. Methodology. The study was conducted in 6 villages of Lower Assam region as
Ma'am, there
shown
is
in the
issue
table.
from your network.
Information on ethnomedicinal uses of plants, the plants parts used, and the mode of administration were recorded consulting knowledgeable informants belonging to two major communities of Assam, Bengali and Assamese. Here you can see some photographs of the respondents. Coming to the result section, the results obtained in the study has been summarized in the table, but I will be explaining them in the upcoming slides. So let's move to the coming slides. So the first species shown in this slide is Diplocyclos palmatus, which is locally known as mala in Bengali. The leaves of this plant is used to treat the juice of the leaves are applied externally on the eyelid, and tender shoot of this plant is consumed as vegetable. So the next is Coccinea grandis, locally known as Dalakucha in Bengali. The tender shoot and fruit of this plant is used as vegetable. The root extract is used as anti-emetic agent, and the chopped leaves are boiled in water, and the warm water is used to treat stomach pain and constipation. So the next is Cucumis maderas paternus, which is locally known as Bontio in Assamese. The tender shoot of this plant is also used as a vegetable. The dried leaves are boiled in water and taken in empty stomach for a minimum period of 15 days to control high blood pressure and high sugar level. Next, a wild variety of Cucumis mellow, locally known as Bon Bangi in Assamese. The seeds of this plant is used to treat fever and headache. The fruit pulp is used as laxative, and the root is used as an emetic agent to cause vomiting. This is actually used when someone consumes by mistake or knowingly poisonous material. Then to cause vomiting, this root is used. The next, Trichosanthis costata, locally known as Batizhinga in Bengali. The leaves of this plant is used to treat skin rashes and ringworm. It is mostly externally applied, and the seeds are used as purgative. The sixth species, that is Trichosanthis tricuspidata, it is locally known as makal in Bengali. The seeds are used to treat constipation and also used as diuretics. Besides, the roots of this plant in combination with Cynodon dactylon is used to make small pills which are used to treat spermatorrhea, a sexual disease. And coming to the discussion section, so we have recorded a total of 6 wild cucurbits under 4 genera of Cucurbitaceae being used as medicinal herbs. However, previous studies have also reported these plants for varied purpose of use, However, the purpose and pattern of use reported in the present study are considerably different, which is shown in the table. So, as time is limited, I'm not going to discuss in detail the tables. So I'll move to conclusion.
Kindly come up. Anand, kindly sum up. We have time for question answers also. So please kindly.
Okay. Just the conclusion part. The species recorded in the present study highlight the importance of the family in terms of herbal medicine. Further investigation may appraise their efficacy and therapeutic potential. And this wild edible and medicinal plants play important role in rural lives. However, indiscriminate exploitation of bioresources may lead to extinction of local population of the species involved. Therefore, it is very important to promote community-based conservation strategy for sustainable use of bioresources. So I acknowledge my supervisor and HOD, and then UGC Non-Net Fellowship, and my colleague research scholars and the village respondents. So these are the references used in the presentation.
Okay, we would like to request anyone, any questions from the audience or who are online.
And any query? Yeah, there is a question, Anand. Please take that question from one of the. Yes, please.
Okay.
Thank you, Anand, for your nice presentation. I have a query. As you have presented that 6 medicinally, ethno-medicinally important cucurbits you told.
Yes.
My question is, what is the status of market demand? Can you tell us?
The market demand, actually, one of the species, which is this Coccinea grandis, the fruits of this plant is also sold in the market. I have seen, but the others I haven't seen in the markets. But they are being used by the local peoples and in those villages.
Okay. So thank you, Anand. I think we will not take more questions right now as there are more presenters waiting in line. I think market demand locally is assessed. It's not commercially exported from Northeast India, Assam region where he is working.
Actually,
So, yeah. Okay. Thank you.
okay, sir. Thank you, sir.
Thank you. Yes. So next presenter, please call on next presenter. Yeah.
We would like to request the next presenter. Importance of invertebrates and their conservation. C. Narasimha Rao.
Sir, now I am presenting my slides.
Yes, you have 5 minutes for presentation, and after that question answers, so be time bound. Can you start sharing your slide, please? Anand, please stop sharing. You have to remove the presenting.
Sir,
Yeah.
is it visible, sir? Hello?
Yes.
Okay,
Yes.
thank you.
You can start.
Respected chair of the present session and the delegates and dear participants. I am Dr. C. Narasimha Rao, lecturer in zoology, Government College for Men Autonomous, Kadapa, Andhra Pradesh. Now, I am going to give a presentation on importance of invertebrates and their conservation. First, the without backbone or vertebral column are known as invertebrates. They occupy about 90% of the total animals present on the earth. For example, protozoans, sponges, jellyfishes, roundworms, flatworms, earthworms, insects, crabs, snails, sea stars, etc. The importance to human beings in many aspects. The insects on land can eat some of the prey and protect our crops. The predatory insects and spiders, they control the pests with none of the health and environmental risk of the chemicals, and they help us avoid the use of pesticides. Some of the bees, crabs, butterflies, flies, and a variety of other pollinating insects. They convert flowers into food for humans, birds, animals, and other invertebrates in the form of fruits, nuts, berries, and some of the seeds. The insects are responsible for the pollination of about 80% of trees and bushes on the entire planet. Fungi and bacteria, as well as decaying and dead debris. Some invertebrates help to remove and clean up the environment. Insects are important in nutrient recycling also by degrading or consuming leaf, litter, wood, carrion, and some of the dung. This is essential for the release of nutrients that are later utilized for the growing of plants. And some of ants and some of the animals, they play a crucial role in the dispersal of fruit and seeds from plants. There are more than 150 species of plants that rely on insects for dispersal of their seeds. The invertebrate also serves as a seafood. For example, we are consuming squids, clams, oysters, shrimps, and also some of the crabs. They also provide nutrition to other animals that include birds and humans. Over 3,000 ethnic groups eat 2,086 species of insects across 130 countries. Insects are therefore an essential source of nutrients to many people around the world. There is also the economic importance of invertebrates. For example, the insects which produce honey, wax, lac, dyes, and silk. These are commercially beneficial. Now we will discuss about the threats to the invertebrate biodiversity. According to a study, invertebrate numbers have declined by 45% on average over a 35-year period during which the human population has doubled. The factors which are causing invertebrate decline are similar to those causing overall biodiversity declines. The factors include loss of fragmentation of habitat, pollution from light, microplastics, and synthetic pesticides, spread of pathogens and parasites, introduction of invasive species, and climate change. There are some of conservation strategies. The main are the basic strategies of biodiversity conservation. There are two types. One is in situ conservation, that means on site. The organisms in its natural home through protection of a group of typical ecosystem by a network of protected areas, biosphere reserves, sacred forests, and sacred lakes. The second one is ex situ or off-site conservation. In this, the conservation of organisms in botanical gardens use conservation stands, gene, pollen seeds, seedling, tissue culture, and DNA banks. Besides this, there are a number of approaches for conserving invertebrate biodiversity, such as protecting the natural habitats, growing of native plants, minimize or avoid the use of pesticides, preventing deforestation, etc. To conserve these valuable invertebrates, we have to develop the research activities which contribute to a better knowledge of species distribution, population evolution, and environmental requirements. Invertebrate species can be protected through the use of modern technology with a broad understanding of the many benefits to humans. Finally, it is also important to create and enhance the awareness among the common people in the preservation of valuable invertebrates. Thank you. Thank you very much, sir.
Any questions?
Yes.
Any questions from online participants or offline participants?
No questions.
Okay, thank you, sir.
Okay, there are no
Thank you
questions.
for giving me this opportunity. Thank you, sir.
Thank you. Thank you. Then we move on to the next speaker. Question. The next speaker is this talk is on a study on the Hydrospheric endangered species of Andhra Pradesh. Presenter is Sachi Devi. Please.
Good afternoon, convener of the conference, the organizing team, the participants, and the delegates. Good afternoon to you, everyone. And my name is Dr. Sachi Devi. Let me present the screen. Is it visible to you, everyone? Is it visible, sir?
Yes.
Madam? Am I audible to you?
Yes.
Yes, audible.
Yes.
Yeah. Yes, fine. I'm Dr. Sachi Devi, lecturer in zoology from SKR and SKR Government College for Women, Autonomous Kadapa from Andhra Pradesh. I'd like to, in my presentation, I'd like to focus on an endemic bird species, that is Jaddans courser, which is a critically endangered species of Andhra Pradesh. I would like to focus on its details and the protective measures taken up by the Government of India in association with the Andhra Pradesh state government. So what is this Jaddans courser? Jaddans courser, its scientific name is Rhinoptilus bitorquatus. It is a nocturnal bird found locally in India. It is only found in India, and especially in the southern India, in the eastern Ghats of Andhra Pradesh, it has been sighted. The bird was first discovered by the naturalist Thomas C. Jerdon in 1848, and on his name it was named as Jaddans courser. Jerdon is his name, and the courser because it is a swift runner. It runs quickly, so that is why the name Jaddans courser. And it inhabits the open patches within the shrub forest in the Lankamaleshwara sanctuary in the Andhra Pradesh. It is the habitat of this courser bird. It is the world's one of rarest bird species and is classified as a critically endangered species by International Union for Conservation of Nature. Let us see the morphology. It is a compact courser with two brown breast bands. It has a yellow base to the black bill. A blackish crown can be seen with the broad buff supercilium. Supercilium is a crown of plumage found on the top of the head. So with a broad supercilium, and an orange chestnut is present. In the it appears like a throat patch. So you can see just below the head, you can see the orange chestnut throat patch. A narrow white crown stripe runs on the top of the head. In flight, it shows a mostly black tail. So it has a black tail and a prominent white wing bar. So these are the morphological features of the bird. And the local name of the bird is Kalvikodi. It is a nocturnal and insectivorous bird which feeds on insects. For over 85 years, the Jaddans courser was thought to be extinct, and it was sensationally rediscovered in the year 1986. So the number is still very less, and it is only around 50 to 150 only the number, and the sightings of the bird is also very rare. So mostly the Government of India, in association with the Andhra Pradesh Forest Department, it is searching for the sightings using the camera trappings. So an increase. So what is the reason for the decrease in the decline the number of the bird, and it is actually that caused on the verge of extinction. So the reasons are an increase in various anthropogenic activities has resulted in loss of shrub jungle habitat where it lives. So especially in the Sri Lankamaleshwara Wildlife Sanctuary, where the species is found, the decreased habitat is the main cause for the irrigation canal construction near this sanctuary. Extensive quarrying and increased agricultural activity resulted in the decreased sightings of the bird. based on let us see the protection measures taken up by our government of India. So based on the long-term research and in consultation with Andhra Pradesh Forest Department, a species recovery plan was prepared in 2010. It has been implemented by the government of India. The main objective of this recovery plan is survey and monitoring through camera trappings to detect the presence of the bird. And also
Ma'am, your slides are not changing. Kindly take note of that.
but I'm changing. So let me come back and is that okay? Can you see the protection measures now? The slide.
Yes, protection measures.
Yeah. So the protection measures taken up by the government of India are based on the long-term research plan and in consultation with the Andhra Pradesh Forest Department. The government of India has launched a species recovery plan in 2010. The main objective of this recovery plan are survey and monitoring through camera trappings in order to detect the presence of the bird and restoration of the shrub jungle habitat, especially in the Lankamareshwara sanctuary area where the bird is found. So in order to restore its habitat, the government has taken up several protection measures. The other protection measures are some of the ongoing activities carried out along the Andhra Pradesh Forest Department. To meet these objectives are establishing nature information and training center. So information and training center has been established for Andhra Pradesh Forest Department officials and also for general public to create awareness about the bird species. Training also was carried out to the for the APFD, that is Andhra Pradesh Forest Department staff, on various latest survey techniques, advanced survey techniques to detect the Jordan's courser and appropriate management of the shrub jungle habitat. So habitat monitoring and mapping was also carried out. Removal of non-native tree species like eucalyptus and biodiesel plantations was also carried out to restore the shrub jungle habitat of the bird species.
Nice,
So these
Dr.
are the
Sachi
protection
Devi.
measures.
Can you please summarize now?
Yeah, because of all these I summarize because of all these protection measures. Let us hope for the fruitful results and the restoration of the habitat may lead to more sightings of the bird species, so that the number of the species which has been declining may increase in the coming future. Thank you, organizing team and the convener for giving me this opportunity and for your patient listening. Thank you.
Thank you, Madam Sachi Devi, for a wonderful bird you have shown and highlighted its conservation. I am just curious before I invite anybody to ask any question. How big is the population of this particular bird? Hello? Got disconnected? Hello? Okay. Any queries from anyone, either connected online or
Yes, sir.
here present in the offline mode? Yeah, please go ahead.
Yeah, sir, I am Anurag Vishwakarma. I'm a PhD scholar. So, ma'am, it's an excellent presentation actually, because this species is really needed for conservation. And I'm just curiously asked two questions. First is what is the current status of this bird, and is there any captive measurement doing by Forest Department or other agencies?
No. There are no such captive growth or measures, and only the bird the number has declined to only 50 to 100, and recently in the recent years it was not even sighted. So that is why more measures were taken up. Advanced techniques were used by the government of India along with the forest department. The cameras were fixed in the forest area, shrub jungle area, that is in the Lankamaleshwara forest range, to sight the bird species. In the recent years, it was not cited by anyone, so that is why it is considered as the threatened species.
Okay, thank you, ma'am.
Okay.
very much, and we will go to the next speaker. This was a very fascinating bird to all of us. I hope we will go for the protection and conservation of this in a larger way with your support, madam. We call upon the next speaker, Priya Kaushik, with Dr. P.K. Khare, analysis of functional leaf rate variation among dominant plant species of dry deciduous forests of central India. Priya, are you there?
Yes, sir. Yes.
Please go ahead.
Please start by sharing your screen.
Hello. Good afternoon, everyone. Respected sir, this is Priya Kaushik. Thank you, convener. Thank you, sir, for giving me opportunity to share my this paper presentation on the white platform. The topic is analysis of functional leaf rate variation among dominant plant species of dry deciduous forest of central India. So it goes like this. I'll tell you the introduction, methodology, field data, data analysis, results, and conclusion. So functional diversity, as everyone knows, it is an essential concept and actually the key concept in the ecological domain because it refers to the relative abundance, range, and value of the functional traits present in the given community or ecosystem. Actually, functional diversity, why it is important? Because it overcomes the limitations of taxonomic diversity. It actually concerns with what organisms do rather than their taxonomic identity, and it provides that how the organism exploit their resources and the functional trait space. And the unit, how to measure the functional diversity, the functional unit is functional trait, the functional trait of the organism. So the functional trait of the organism is what? The traits of the organism which impacts the organism's survival, the growth, and the reproduction. So we can have the leaf traits, stem traits, root traits, which creates a link between ecosystem processes and plant physiology. Thus offer an effective tool to study the global change on vegetation dynamics and ecosystem processes. This leaf functional trait is important because it is associated with ecosystem level processes, species coexistence, plant growth, and plasticity. So the present study focuses on how the plant functional traits helpful in predicting the patterns of community assembly and describing species contributions to ecosystem processes. So I have divided my study area in the dry deciduous type and the moist deciduous type. So the first site is Sagar, the second is Betul. It is highly diversified deciduous forest covering the unwritten topography of low-rising hills. You can say the temperature, the highest temperature, maximum temperature is 45 degrees Celsius during the month of May, June, and the annual rainfall is 1240 millimeter. And the forest communities are dominated by Tictuna grandis, Butea monosperma, Holoctelia, and Eudendron glaucum. Here the second site is Betul. Here you can see the climate is monsoonal with seasonal rainfall pattern, maximum rainfall of 850.2 millimeter, and the maximum temperature is 40 degrees Celsius and the minimum is 15 degrees Celsius. Again, the community is dominated by Butea monosperma, Chlorella sweetenia, Legeristomia, and many more. So sampling design, how we collected data in the field. We have covered 21 plots, that is 50 meter to 50 meter in the ranges of Betul and the Sagar. So what we for our easy method, what we do, we divide it into 25 to 25 subquadrants, and then I have selected 9 functional traits, that is specific leaf area. This is the most important trait, functional attribute we can say. Why? Because it tells you about the main leaf area. Because if leaf area is low, so what happens? जितना कम लीफ एरिया होगा उतना ही उसको हर्बीवोरी उस अटैक से बचेगा वो, और you can say it provides the resource use efficiency of the organism. Okay, And the second major trait is LDMC, leaf diameter content, leaf size, length-width ratio, leaf thickness, and basal area. Okay. So what we do, we chose the healthy, robust, disease-free plants selected for trait measurements. Traits were measured over at least 10 individuals per species. For analysis, mean trait values were used per species. Okay, so this is this I have already explained, and then frequency, density, abundance, and basal area. These are the major. Why? Because frequency, what it depicts the numerical distribution of a species in the area of per unit area. What density determines? Density determines the numerical strength of an individual of a species per unit area. And abundance, it determines the numerical strength of the individual of a species in the area of its distribution. So these are the distinct. So these are my data.
Can you hear me? Yes, sir. Okay. So can you please just skip the methodology and come to the conclusion?
Yes, sir. Yeah, sure. So you can see the results. The site one, the Sagar. Here you can see that it is dominated. The first comment is dominated by Tectona grandis, which is 100% frequency. Again, the Holothelia integrifolia with 90%. Okay, on the second side, the Betul. Again, the Tectona grandis is the major dominant species were Tectona grandis only. Here we can compare the functional trait values of among the two sites. So SLA, we can have the SLA value. The Tectona, again, it has performed well in both the sites, and the less you can say the Acacia fistula and Gliricidia sapium. It is the rare species which have less values of mean trait values. Correlation, we can see the correlation matrix. Here we can depict that that leaf dry matter content is positively related with leaf size and leaf thickness, and it is negatively related with specific leaf area. So these are my results. Okay, okay, I'll skip. I'll come to the conclusion. Interspecific trait comparisons may have numerous possible outcomes. The plants have similar habit, and living in the vicinity of each other may exhibit a very different set of traits. Functional diversity influences various ecological processes and offers several ecosystem services, but its effects on the intensity of ecological services are controversial. Thus, to understand this more clearly, firstly, functional diversity should be further studied in different ecosystems. It is essential for predicting patterns of community assembly, as well as for describing species contributions to ecosystem processes. So, further studies are required in the field of ecology due to their importance in understanding and management of forest ecosystem. Thank you.
Thank you for your beautiful presentation. Any queries from any online participants first?
My question is, you told that Visual monosperma is a dominant tree species.
Yes, sir.
It is true.
Yes, sir. Actually, Sagar and Betul. Sagar is a dry deciduous forest type, as you know, as we consider the champion and said classification. So the Tectona grandis, Tectona and Butea, and you can say Diospyros. These are most dominant species in the Sagar area.
Okay, but my point of view, Tectona grandis and other major species are also dominating. But
yes,
as
yes.
far as Utilisation monosperma is concerned, it is not dominating species in any way.
Sir, I think it is co-dominant species. Yes, we can say it is a
very good role in the forest.
Yes, sir.
Okay, thank you.
Thank you, sir.
Hello, we have very good presentation. I am
Thank you, sir.
Faculty of Forestry, Gurgaon. I have very simple question. Is this herbivory is good or bad for the ecosystem function?
Sir, sometimes it is good, sometimes it is bad, we can say, because if we talk about the invasive species, okay, so that time what herbivory plays the role. Herbivory उसको सर वो मतलब एक्चुअली क्या होता है कि जब इन्वेसिव स्पीशीज होती है तो उसकी लीफ थिकनेस का जो एसएलए होता है वो बहुत कम होता है. अगर एसएलए कम रहेगा तो उसकी लीफ थिकनेस और वो जो चीज लीफ डायमीटर कंटेंट हाई होते हैं जिसकी वजह से हर्बिवोरी उसे मतलब दैट इज लेस अट्रैक्टेड टू हर्बिवोरी, सो उसको चांस मिल जाता है आगे बढ़ने का, तो वो इन्वेट कर जाता है प्लांट, और फिर वो हार्मफुल होता है बिकॉज इट डिक्लाइंस द नेटिव बायोडाइवर्सिटी। तो उसकी वजह से इस टाइम पे हम देखेंगे कि हर्बिवोरी सही नहीं है, लेकिन कहीं पे अगर वहीं पे वो इन्वेसिव प्लांट को अगर वो खा लेता तो शायद वो हेल्पफुल हो जाता। सर वी कैन प्रेडिक्ट इट दैट वे।
How it is helpful? Let's actually-
यस सर, सॉरी
How it is helpful?
सर इट इज हेल्पफुल इन दैट वे, अगर वो प्लांट इन्वेसिव है और वो अगर हार्म कर रहा है नेटिव बायोडाइवर्सिटी को, तो सर उसको वहाँ पे हर्बिवोरी अगर अटैक करता है, अगर उसके लिए वो मतलब, तो फिर अगर वो उसमें उसकी डाइवर्सिटी को कम कर देगा तो नेटिव बायोडाइवर्सिटी हमारी बची रहेगी, इसकी वजह से यहाँ पे वो अच्छा रोल प्ले करेगा।
Okay, nutrient cycling is very helpful. Okay, it increases the diversity of the microorganisms.
Yes sir.
A lot of nutrients are being added to this one. Thank you.
Okay, thank you sir, thank you.
For your presentation, we will move on to the next speaker now.
Thank you sir.
Thank you. Okay. So next speaker is Anurag Vishwakarma and Avdesh Kumar. The use of regular monitoring for bird species to identify potential habitat in forestry for AV tourism expansion and upkeep wildlife.
Okay. So good afternoon to all. Myself Ankita Dobaria, and I'm pursuing PhD in Saurashtra University, Rajkot, Gujarat. And today I'm going to present on a phenogram cluster-based seasonal analysis of the marine actinomycetes from a pristine sea coast of Shivrajpur, Dwarika, Gujarat. So first of all, what is the actinomycetes? Actinomycetes are a diverse group of bacteria in terms of the different metabolites and enzyme production. And marine habitat serves a rich source of the microbial diversity with a unique physical, biochemical, and adaptation feature. And especially halophilic actinomycetes reflect the wide diversity at genetic and phenotypic levels. And the diversity analysis is an important part to study the ecological relationship between different species. So in a coastal region, Streptomyces and Nocardiopsis genera are the dominant groups, and this present study describes the phenogram-based diversity of alkalophilic actinomycetes of the different seasons. So first of all, the marine actinomycetes strain were isolated from Kachigadi coast near Dwarika, Gujarat, and during the pre-monsoon, monsoon, and post-monsoon seasons. And the metabolic characterization was carried out based on the different sugar utilization test and biochemical test. And then phenograms of all three seasons were isolated and constructed by the UPGMA algorithm and Jaccard similarity coefficients, and the past software used for the construction of the phenogram. And the term phenon indicates the cluster in a phenogram. That indicates the greater number of clusters with higher phenotypic similarities and then a lesser number of the diversity. So then after that studied the phenotypic diversity of all three seasons in a comparative way. So first of all, these figures represent the phenotypic and metabolic characterization in terms of the positive percentage of the pre-monsoon, monsoon, and post-monsoon seasons. And these isolates were characterized based on the different biochemical tests such as urea hydrolysis, citrate utilization, triple sugar ion test, gelatin hydrolysis, oxidase test, etc. And then after that, the person correlation analysis was carried out to confirm the survival capability of the isolates in different conditions. And the past software algorithm were used for the creation of the correlation charts. And this chart displaying the relationship between the selected physicochemical parameter and corresponding biochemical test. So in this figure, deep blue color indicate a strong correlation, while color shifting toward a red indicate the regression. And then cluster analysis represent the phenotypic characteristics of the microorganism. So in this figure represents the phenogram of the pre-monsoon isolates and displayed the greater phenotypic and metabolic distinctness is only single cluster between the KS5 and KS13 with 100% similarity, and remaining other isolates form lesser cluster. After that, this figure represents the phenogram of the monsoon season. So in the phenogram of the monsoon season, so four cluster in the first cluster formed between the KM5, KM7 and KM8, and second cluster formed between the KM10 and KM11, third cluster formed between the KM11, KM13, and fourth cluster formed between the KM3 and KM6 with the 100% similarities. This phenogram indicates the post-monsoon seasons isolates, in which total 3 clusters were found with 100% similarity. First cluster formed between the KW3 and KW13 isolate. Then second cluster formed between the KW9 and KW26. And third cluster formed between the KW7 and KW24. Then some other subcluster also found with lesser similarity, which is present less similarity between the different isolates. Then this phenogram indicates the comparative study of the all three season isolates. So this figure reflects the total 7 cluster with the 100% similarities. this is the summary of the presentation. So actinobacteria were first of all isolated during the different seasons from the Kachikati coast near Dwarika, and in the phenogram of the monsoon season, total 4 clusters found with 100% similarity, and in the post-monsoon season, total 3 clusters were found with 100% similarity, and other sub-cluster also found with lesser similarity. In the phenogram of the pre-monsoon season, 1 cluster found with 100% similarity, and in comparative studies, the total overall 7 clusters were found with 100% similarity. So pre-monsoon isolates have a greater metabolic and phenotypic diversity compared to the monsoon and post-monsoon season. So this study established the pre-monsoon season has a higher metabolic traits and diversification. This is the references of the presentation.
That's fine.
That's fine. Yeah, Ankita.
Okay, sir.
Any query? Any quick query for this presentation by Ankita Dubai? Any online participant wants to ask any question, please? There is one.
Ankita,
Yes, sir.
can you say which factor is responsible for this variation in different seasons, pre and rainy and post rainy season? What do you think?
Yes.
Which factor is responsible for this dissimilarity of your isolates?
Sorry, sir, I can't listen properly.
You said that there are variation in the similarity rate. Pre-monsoon season,
Yes, sir.
monsoon and post-monsoon.
Yes, sir.
Sometimes you are getting 4 cluster is similar, sometimes 3 is similar, sometimes. So, what makes these isolates or group of microbes different? Why this variation you are observing in your experiment? What is the main reason?
Sir, because of the this study carried out for the diversity analysis in different seasons. So that is the main purpose in a species variation, and some similarities were found in a total in all 3 seasons. So that is the main factor behind the similarities. It is nutritional availability, I think.
You think, or have you tested some nutritional availability? Any test you made that nutritional availability is more in this season or any season? In your study?
Sorry, sir, I can't listen properly.
Have you made any experimentation or any such things which show that this nutritional availability is responsible for this variation?
No, sir. No, sir.
I think you can do it. You can do it in your next experimentation.
Yes, sir. Yes, sir. Thank you, sir.
For availability of nutrition and correlate with the dissimilarity of your isolates.
Yes, sir. Thank you, sir, for your suggestion.
You have some good suggestion. Very nice suggestion from sir.
Thank you, sir. Thank you, ma'am.
I would like to include you mentioned Parsons correlation somewhere. It's Pearson's correlation to check it. Okay. So thank you very much. We go to the next speaker.
Thank you, sir.
We have some little bit of jump as there is a request from Alpana Ram and Animesh Pandey. Alpana Ram will this particular presentation. Microspores of Fullerena and license extract for hepatic malignancy. Are you there?
Hello, and am I audible?
Yeah, please go ahead and start your presentation. Thank you.
Okay, respected chairperson and the convener and the chairpersons of this technical session. I am Dr. Alpana Ram from Department of Pharmacy, Guru Ghasidas University. I wish to present my screen. Is it visible?
It's coming up now. Just
okay.
yes, now
So my topic is My topic is microspheres of Holar henna antidicentrica extract for hepatic malignancy. As it is well known that hepatocellular carcinoma is the ultimate result of chronic infections with hepatitis B and C viruses, alcohol consumption, and aflatoxin exposure. It has a poor prognosis because its progression is very rapid, and it is usually detected at a very advanced stage. And it is the fifth most common type of cancer globally. Its development is a multi-step process involving sustained inflammatory damage, including hepatocyte necrosis and regeneration with fibrotic deposition. So I have decided to take the bark of the conifer tree, that is Holar henna antidicentrica, belonging to the family Apocynaceae. It has been included in the British Materia Medica since a long time, and it is also being used indigenously for the treatment of dysentery and diarrhea. But several Indian tribes have used it in other diseases like anemia, epilepsy, and cholera. So it has also been reported in Ayurvedic and Unani system of medicine as an enthalmentic. So and it is also used in the treatment of skin diseases. So I chose that it should be useful for the treatment of hepatocellular carcinoma also. So we have formulated the microspheres out of it, because most of the conventional oral dosage forms, they have a number of disadvantages. The monolithic microspheres were prepared where the drug is distributed uniformly in the matrix system as a molecular dispersion of particles without any distinct outer layer. And these microspheres, they have several disadvantages, and they can be made as free-flowing powders using non-toxic biodegradable polymer, that is chitosan. And this is the extraction process. The bark was collected from Amarkantak region. It was dried and finely powdered, macerated with 70% methanol for 3 days, and the solvent was removed, and ultimately it was lyophilized. The residue was lyophilized. The obtained extract was used for the preparation of microspheres. Now, chitosan was dissolved in 1% of acetic acid solution. It was sonicated for 10 minutes to remove the air bubbles. The extract was dissolved in water, and it was added to the previous solution. And it was added dropwise to the oily phase at room temperature with continuous stirring. And glutaraldehyde solution was added, and the studying was continued for 1 hour. And suspension of chitosan microspheres in paraffin oil were obtained. They were allowed to stand till the microspheres settled down. The supernatant was removed and washed with petroleum ether and acetone to remove the traces of oil. And finally, it was washed with water to remove the excess of glutaraldehyde, which was used as a cross-linking agent. Then I have studied the entrapment efficiency and percent yield, and actually 3-level design was used, and 14 formulations were prepared. And out of these 14 formulations, F11 formulation was selected as the optimum formulation, which was based on the highest percentage yield and the highest entrapment efficiency of 69.29%. Apart from this, the FTIR studies, photomicrographic images, the SEM, TEM, and DSC, and other micromeretic studies were conducted, and the in vitro release in the phosphate buffer pH 7.4 was carried out. And then the human hepatoma cell line was used for in vitro studies, and the results showed that 80 microgram per ml concentration of the extract exhibited 17.1% cytotoxic activity of the cells after 48 hours of cell exposure, and the growth inhibitory 50 value of the plant extract against Hep G2 cell lines were studied. And apart from the extract, the microspheres were also studied, and the results showed that 80 microgram per ml exhibited 16.23% of cytotoxic activity in the hep G2 cell lines. Then the hepatoprotective activity was studied using, it was compared, the methanolic extract was compared to the microspheres, and generally paracetamol is antipyretic in therapeutic doses, but it produces fatal hepatic necrosis in man, rats, and mice with toxic doses, and those up to about 2 gram per kg was used, which produces depletion of hepatic glutathione. So paracetamol caused significant elevation of the levels of SGOT, SGPT, the serum ALP, and bilirubin. So to assess the hepatoprotective activity, rats were used, albino V star strain rats were used, and they were administered with 200 milligram per kg as well as 400 milligram per kg of the methanolic extract given orally for 1 week, and the microspheres were given in a dose of 100 milligram per kg as well as 200 milligram per kg, and on the 5th day of administration they were given paracetamol at a dose of 2 grams per kg orally, and blood was withdrawn by cardiac puncture and tested for SGOT. So all these tests were done, and the animals were sacrificed, and livers were excised and quickly stained with hematoxylin and eosin for histological architecture. The sections were cut and observed under microscope. The first one shows a normal liver with a prominent central vein, and normal hepatocytes are there. The blue stains are the nucleus, and the pink one is the cytoplasm. So here we see the second in the second one we see that this is paracetamol.
This point to me kindly say that.
I cannot hear, sir. Did you say something?
Just in one line, can you summarize now your talk?
Okay. So I have compared it with silimarin, which is used as a hepatoprotective, and I have seen that the microspheres of Holarhena antidicentrica, they are protective for hepatocellular malignancy.
Thank you.
So all these studies, they reveal that the Holarhena antidicentrica bar extract microspheres have proved to be protective against hepatocellular carcinoma.
Okay, very nice. Any queries? Good queries. There is one from Madam here.
Okay, I have one question, Dr. Ram.
Yes.
How the treatment for this drug actually? Before sacrificing your animal, it is a 6-month study, or it is a 1 month, or it is a 15 days?
Yeah. For 1 month they were given, and the animals were sacrificed after 1 month.
After 1 month, okay.
Yes.
It was a complete 1 month study. And,
Yes.
and in this drug, what is the bioactive molecule you find in this drug actually?
Conesin, sir. Conesin.
Conesin. Okay.
Yeah.
Thank you.
Thank you. If there is no other query, I think I will just mention here.
Okay, one more question.
One more is there. Okay, so just a small suggestion and a
Yes, sir. Yes, sir.
a small suggestion on the name of the plant species you have mentioned, Holarhena antidicentrica, also called as Pucci. I think we check the spellings of the Holarhena and also the specific epithet. It changed now to pubescens.
Yes. Okay, sir. Thank you, sir. Thank you.
Holarhena pubescens. Please just check it. Thank
Yes,
you
sir.
very much. And we move on to the next speaker.
Excuse me, sir. Sorry for the interruption.
Yes.
I'm Anurag Vishwakarma, sir. Actually, I am ready, sir. Sorry for the technical issues, sir.
No
right
problem.
now, I am ready, sir.
Okay, okay. So please go. Okay, we call upon Anurag Vishwakarma, and please start presenting. You are welcome.
Yes, sir. I'll be presenting my screen. Sir. Is it visible, sir? Is my screen visible?
Yes, 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 yes.
Okay, thank you. A very good afternoon to all the chairperson, co-chairperson, faculty members, my dear friends and participants. Uh, Myself, Anurag Vishwakarma. I'm a PhD scholar in Department of Forestry, nearest Itanagar, Arunachal Pradesh. I'm doing my PhD under supervision of Dr. Avdesh Kumar, who is associate professor. So today I'm going to present a small work from my PhD objective, that is the use of regular monitoring for bird species to identify the potential habitat in forest trails for AV tourism expansion at Pake. So AV tourism means the tourism based on the birds. So AV tourism is rapidly becoming a solution in today's biodiversity conservation battle by minimizing exploitative behavior of the environment. The conservation work on Arunachal Pradesh has focused particularly on the bird species. Why? Because of the Arunachal Pradesh is a part of Eastern Himalaya landscape and is one of the biodiversity hotspot in India as well. Eastern Himalaya reported 831 avifauna species out of 1,341 species reported from Indian subcontinent. And Arunachal Pradesh consists of around 750 avifauna species, and out of 296 avifauna species reported from Pake Wildlife Sanctuary only. Hornbills are the major attraction species in entire Arunachal Pradesh because they have some specific value. Here, local people are using beak of great hornbill in their traditional headgear from last many decades to show their power, wealth, and prosperity. That's why their population is going to be decreased. Pake Tiger Reserve has poor record on tourism, and previously no such study has been conducted on avi tourism based and avifauna population. So by focusing of the bird species, the present study, we attempt to provide an initial description of the bird species found in the selected nature trails with unique and high abundance of bird species. My study site is Pake Wildlife Sanctuary, which is situated in Arunachal Pradesh, Pake Kesang district, and it is having 861.95 square kilometer. It is also a tiger reserve proposed in 2002. The climate type varies from tropical to subalpine. As you can see, the altitudinal range varies from 150 to 2040 millimeter above sea level. And the average rainfall is 2500 mm, and average temperature is 12 to 34 degrees centigrade. For my survey, I have done point count distance sampling method, and in which 124 sampling points were laid in between 500 millimeter of distance for the overlapping of the bird species. And I have done, and the period of my study was 2020 and 2021 consecutively, pre-monsoon and post-monsoon season. So why pre-monsoon and post-monsoon? Because in Arunachal Pradesh, the lands, the climate is vary because there is no summer season from April onward. The rainfall has started, so that's why I have taken this pre-monsoon and post-monsoon replicates. So in my results, I have selected total 9 nature trails. Previously, there were only 7 nature trails were presented, and these 2 nature trails, this white one and this white one, I have added after my study. And the longest nature trail is this inside the core area, which is this Dikroy to Panchali. This is 13 kilometers from the longest trail, and this is the smallest nature trail I have selected in my study area, which is 3.5 kilometer. So during my study, this fourth number, Dikroy to Panchali, this is the longest trail, 13 kilometer. I have identified the highest number of species in this nature trail and the lowest number of the species in this Python to DG nature trail, because this is the 4 to 5 kilometer of nature trail, and just because of the probability of the dense forest, so that's why the species number of species is very low here. Same number of individuals are higher in the West Bank to Khari. West Bank is the entry gate of Pake Tiger Reserves, and from there most of the tourists are going for the bird watching. So that is the 12 kilometer of long tail, and the number of individuals are very high just because of the probability of sighting of species is very because the forests are like open forests. So the probability of sighting species is very, very high. That's why the number of individuals is higher. And this Python 2 DG, this is also again uh, just because of the dense forest, the probability of sighting species is very low. So that's why the number of individuals is again low here. And the Shannon Diversity Index, I have reported the highest diverse species in the TP, and TP is the newest nature trail, which is I have added after my study. So there I have got the highest number of diverse species, and this Python 2 DG have the lowest diverse diversity. And overall Shannon diversity in 2020 and 2021, Nikroi and Panchali, which is the longest nature trail, is the 13 kilometer. So have the highest diversity, and this Python 2 DG have the lowest diverse nature trail. So during my study, in these 9 nature trails, the most abundant species I have got this black bulbul, this is scarlet minivet, and this mountain imperial pigeon. So these are the highest abundant species in both the all the 4 replicates and 2 years. These are some of the important high abundant species. This is black bulbul, this is blue-winged Manila, and this is the targeted and focused species, bright kingfisher, and this is rufous-necked hornbill, which is endemic to Northeast India only. So most of the people are coming to watch these hornbills and these bright kingfisher only. So in conclusion, in our study, Oriental pied hornbill, wreath hornbill, great hornbill, and bright kingfisher are the major focus bird species we have cited. And see, AV tourism, it can be a potential livelihood because people are getting direct and indirect benefit from this kind of tourism business. And obviously, if they will get something from conservation of one species, so they will definitely conserve the entire wild flora and fauna species. And in near future, I'm hoping that Arunachal Pradesh can be a rich and unique biodiversity area, and it can be a birding hub for the wildlife lovers and bird watchers. These are my references for this presentation, and I would like to thank these following persons, especially my supervisor, Dr. Abdesh Kumar, who supervised me or support me and guided me very well during my study. Thank you.
Thank you very much, Anurag. Wonderful work. Any queries from any? Yes, there is one query from Tiwari here.
Anurag,
yes, sir.
You have done wonderful work in Pathway Wildlife Sanctuary as well as in Central India also. My simple question is, first of all, I must congratulate you because you are our alma mater. So we are very happy that you are doing very good work in Arunachal Pradesh also. My simple question is, because you have worked in both the landscape, in Central India as well as the Northeast Himalayas, what is the difference in the diversity of the birds in both these landscapes?
Sir, obviously there is a diverse place because Northeast India and where I am working, it is a biodiversity hotspot, and the number of species are obviously higher. As I told in my introduction, that India is having 1,341 species, in which 831 species are found only in Eastern Himalaya landscape, and in Arunachal it is around 750. But in Central India there are lakh number of species just because of the lots of habitat loss, anthropogenic pressures, and disturbance. So in Central India landscape, species can be more, but just because of the lots of disturbance, the species number are like there.
What about this grey hornbill?
Grey hornbill.
Yeah.
Sir, I have not done any kind of work on grey hornbills in there, but on my on the basis of my experience, I can say.
Another question from parties.
Okay.
Sir, good afternoon. I have a question.
Yes.
Am I audible?
Yeah, you are.
Sir, does trail length and disturbance Like you have tested across different trails. So did the trail length and disturbance have any effect on species composition?
Sorry, Fergus, I couldn't get you.
Sir, the trail length, did you compare the trail length and disturbance with species composition? So does the
No.
longer trail have more species or lesser trails have better species? Did you test that?
Fergus, I'm sorry, but I couldn't get you properly.
I think you have the answer in your figures already. It is there. You have shown the longer bars and smaller bars with respect to different things. Anyway, next question, please.
Hello, Anurag. I am Garivan Tiwari. Hello.
Yes, ma'am.
Anurag, आपने बताया कि you have collected data in 2020 and 2021, and
Yes,
you are
ma'am.
talking about the faunal heavy ecotourism. You have used that term. Hello.
Yes, ma'am.
और जैसा हम सब जानते हैं कि 2020 and 2021 दोनों जो है ये कोविड की एरा थे, तो इस पे आपने कैसे मॉनिटर किया? यहाँ पे तो इकोटूरिज्म का स्कोप जो है it was very limited, ना?
Yes, I know, ma'am. But my study is based on the future work, ma'am, not from the current scenario. Because we all know the current scenario, but in future, this AV tourism can be a good source of income and good livelihood income generation for the local people. So that's why I have focused this work for the future purpose only.
You have to mention some suggestions also, like training of the local community regarding the ecotourism aspects and all these.
Yes,
so you
yes,
have
yes.
incorporated all these things in your studies also.
Yes, I already did, ma'am, all that.
Okay, one more query. Okay, no? Anybody online who would like to ask any question from Anurag? Okay, घूमने के बहाने ऑलरेडी बहुत सारे थे, तुमने एक और ढूंढ लिया है. So I think you have to strike a balance कि ये sustainable होगा कि नहीं biodiversity के लिए ecotourism, increasing ecotourism everywhere all the time, is it good or not? It's questionable under discussion. But you have fascinated all of us with the bird diversity there. I would like to personally interact with you later on, and I think we have people from Marist here sitting. Remember us with your supervisor, Abdesh Kumar. Thank you very much, Anurag. We have from organizer side one message that we will be breaking for lunch now, and we will be back after some time. How many minutes, please? Half an hour, 30 minutes. So those connected online also, please enjoy your lunch at your venue, and we will be back after 30 minutes. And that will be 250 to 220. 220, that's it.
Yes.
So 220 we will be back. Thank you very much for your patience. They are having a little bit of technical problems. We apologize for that. Thank you all.
Thank you, sir.